Hi everyone. Before we begin, I'd like to acknowledge that UBC's Vancouver campus is located on the traditional, ancestral, and unceded territory of the Musqueam people. And as someone personally who is not Indigenous to these lands, I do want to express my deep respect for the Musqueam as the original stewards of these lands, going back for thousands of years. I know that this land acknowledgement is too brief and too insufficient in many ways to really capture the depth and the historical weight of what I'm trying to acknowledge. That said, I do want to, in this brief moment, bring attention to the fact that many Indigenous voices and perspectives have been silenced or erased entirely, as UBC has grown in the past century on these lands. With that in mind, I would like to express my own commitment and extend an invitation to all of us to be proactive in seeking out and in learning from and elevating the Indigenous voices and perspectives that are among us in the present. I also have the honor of introducing our speaker for today, who is Dr. Ted Robles, a professor at UCLA, where he's currently serving as Vice Chair of Graduate Studies and also runs the Relationship and Health Laboratory there. I don't actually think I've met Ted in person before, which is kind of crazy to me, given how much overlap there really is in what we do and what we're excited about. So I'm especially just really thrilled that he could join us today, and because I've known about and followed his work for years. His work is incredibly interesting and broad-ranging. He's done so much work on a number of health-relevant biological processes, including autonomic and cardiovascular responses, HPA stress reactivity, immune responses, inflammatory responses, all in the context of our daily lives and in relationship to stress and to social support and to close relationships across our lifespans. So given the breadth of his work, it's perhaps not surprising that his work has been published in journals that really span the entire range of our field, including reviews that he has in Psychological Bulletin, in American Psychologist, Current Directions in Psychological Science, and empirical work that he has in psychosomatic medicine, archives in general psychiatry, health psychology, developmental psychology, and really so many more. But in addition to his impressive contributions to shaping knowledge and to shaping research in our field, he's also really putting his research on social relationships and health into action. So he's the Engage Well Pod co-leader at UCLA's Semel Healthy Campus Initiative Center, and I think he'll be telling us a little bit about that work today. So without further ado, I would like to hand the virtual floor over to Ted. Welcome,
Great.
Ted.
Thank you. Thank you very much. And as I transition to sharing my screen, I also want to highlight that we are also on traditional and unceded land of the Tongva peoples. And one of the things that's sort of really fulfilling recently is I have a student, I'm not going to talk about this work, but we have a student where we are looking at health outcomes in Native peoples. And so it's really exciting to be able to kind of like try to take a couple of steps towards giving back, given so much that was taken away. Can you guys see the screen okay? Okay, awesome. So thank you for the introduction, and I'm actually really excited sort of seeing everybody's names here, like especially, you know, for those of you who I don't know, but I've seen your names. Those of you I do know, it's really wonderful to see you again, and it's a real honor to be invited to talk with you about my work. So this is going to be a two-part talk. So I'm going to be talking about research from families to immune cells first. That's really my sort of core research program that we've been doing for, you know, the last, 15, 20 years. And then I'm also going to share some of the newest work from that with you. And then after that, I'm going to do a brief overview of work that I'm involved with on our campus to improve health and just sort of seeing the kind of research interests that I know many of you have. I'm hoping that this will be of interest to you as well. With the empirical work that I'm going to talk about today, I just want to emphasize that this is all work that has a lot of potential, but I'm hoping that other people will start to take up that potential in the sense that, you know, our work is consistent with other findings, but it comes from cross-sectional designs. You'll see we have relatively small samples. And so, you know, really a lot of this is what I view as, you know, hypothesis generating work. Needs replication, needs extensions into different contexts and different populations. I'm also going to begin at the end and just sort of acknowledge some folks first, especially since I'm going to transition into sort of a second talk. The work that I'm going to talk about was supported by the William T. Grant Foundation, an RO3 from an ICHD. My core collaborator through all of this has been Rena Rapetti, as well as Steve Cole, and really wonderful postdocs and research associates and other collaborators who are listed on that slide as well as grad students and staff that were involved in the project that I'm going to spend most of my time talking about. Okay, so if you have studied stress at any point in the last half century, you're going to be familiar with this very simple basic model that exposures to stressful events are going to lead to stress responses that then impact physical health. And likewise, if you've studied the role of social networks and social support or close personal relationships in health in the last half century, you're familiar with this basic model, which is the stress buffering model from Cohen and Wills. It's the idea that other people can serve as a buffer that protects us from the negative impact of stress exposures. And in that original model, the idea was that our social relationships, our social support from those relationships can reduce the impact of stress exposures by changing how we think, changing our appraisals, changing how we feel. So that could be changing our affective responses, and then also changing our physiological responses. So, and this should be very familiar. It's basically a higher resolution reproduction of the Psych Bulletin paper that we're all probably familiar with from a while back. What I'm going to do today is show you how we've been trying to unpack the biological pathways that might explain social buffering, and in the process kind of generate some new directions for research on social buffering, particularly in family relationships. And I'm going to focus on warmth and conflict in parent-child relationships. I'll pivot to some of the work we've been doing on relationship closeness and romantic relationships, and then I'm going to pivot back to parent-child. But that is kind of the area in which I work, and working with Rena Rapetti on a lot of this is really, I've moved from a couples researcher to sort of an armchair family researcher, I guess we'll say. I'm also going to talk about the links between our social world and the molecular biology of immune cells. So before getting into some of that data, I do want to take a brief detour. We're going to zoom in on this figure, and I want to talk about links between our social world and what happens in immune cells. And what this figure is supposed to depict is, we have our social world. It gets transduced by our brain. Signals about that world get sent to our periphery, to our body. So through the HPA axis, through hormones like cortisol, through the sympathetic nervous system, through norepinephrine and epinephrine as neurotransmitters. These little guys here are supposed to be bacteria. These are immune cells that are responding to that bacteria, and among the things they do is they produce an inflammatory response, which I'll talk about in much more detail. So let's zoom in on that for a second. So here we have inside the cell. I want to first focus on this part right here, which is we have a bacteria. This immune cell detects that you are exposed to, that there's a bacteria right next to it. And what happens within this immune cell is a cascade of events that eventually leads to the transcription of our genetic code into mRNA, which is then translated into proteins like chemical messengers in the immune system. These are proinflammatory cytokines, interleukin-6, interleukin-1 beta. If you've read anything about inflammation, this will be familiar. They have numerous effects in the body to help kind of promote the immediate response to infection and injury, as well as recruit help in fighting off those invaders. In addition, here's those neuroendocrine and autonomic contributions. So for instance, norepinephrine from the sympathetic nervous system can directly stimulate this cascade on its own. Cortisol can inhibit this cascade. That's why there's this little line here. One of the key players is NF-kappa B, nuclear factor kappa B, which when this is activated, say by bacteria detection or norepinephrine, it activates and moves into the nucleus where it promotes the transcription of our DNA code into mRNA. So here's a closer look at that process. We have NF-kappa B. It binds to a particular region of this gene called the promoter region. When it does that, it initiates the process of transcription. So say this is an IL-1 gene that then transcribes the mRNA strand that codes for IL-1. Here's an example where we have transcription that's coding for, or we have transcription of the gene that codes for IL-6. Here's a little IL-6 mRNA. And then here's cortisol. And what it's doing is it's actually binding to this promoter region, and it's preventing transcription. So the way that you can affect how a cell works is by affecting what genes are turned on, what genes are turned off in real time, and either in promoting transcription or preventing transcription. So if we zoom back out, then that mRNA gets translated into the proteins that are often studied in circulation. So that's like I don't know how long that was. That was like the 2-minute version of inflammation. But I wanted to kind of explain the role of gene transcription factors that can promote or block that process, because that's relevant for what I'm going to talk about next. So I'm going to focus on 2 mechanisms today, which we've been able to explore by measuring gene expression in immune cells, which has opened up new avenues for examining social buffering. So I'm first going to talk about links with pro-inflammatory signaling, followed by immune cell aging. And then after I talk about some potential health implications of that work, I'll sort of turn to future directions if this is if these are plausible pathways. So the gene expression work I'm going to talk about comes from what we call the UCLA Families and Health Study. This was a study that we designed to look at links between the family environment and upper respiratory infection susceptibility and severity in a small sample of families in the community. This was supported by the William T. Grant Foundation. When Rich Slatcher, who's now at University of Georgia, was a postdoc working with me and Rena, we worked together in putting this grant together, and he was really instrumental in that. We also had other funding from the campus, and then eventually for some of the cellular aging work and RO3 from NICHD. Actually, let me just briefly say something about so we have 47 families. They all had to have a child between the ages of 8 and 13. If you have kids between that age, and kind of remarkably, I do now, which is kind of, it blows my mind. But one of the things we noted is like, if there were 2 kids who were eligible, we didn't want to like just randomly pick one. We enabled like some siblings who were within the age range and met criteria to participate. Certainly that would have been what we would have done in my family, or else I would not hear at the end of it. So these kids were an average of 11 years of age. You can see the sample here. If you do family research, you know that dads are not exactly the most willing to sign up for studies. So we had slightly fewer fathers than we had participating mothers. Both parents were encouraged to participate, but they didn't have to. And then you can see the demographic breakdown there. So this is overrepresentative of white or the demographics for white folks in the Los Angeles area. But I'm always sort of impressed about what we were able to do in terms of other race ethnicity groups. We purposely recruited people from zip codes that were not Beverly Hills, or that were not like right over here is the Holmby Hills, where sometimes we have fundraisers for presidential candidates and stuff. We took a lot of steps to make sure we were trying to recruit folks who were at least not super high income, and you can see the median individual income there, which isn't that easy to live on in Los Angeles. So the families began our study with a visit to their home, where we interview participating parents and children using the UCLA Life Stress interview that was developed by Connie Hammond. We can assess ongoing chronic stressors over the past 6 months in a number of domains within the family, within work, and we can also look at episodic stressors. We then had this middle portion here was really the core part of our study. This was a 8-week-long daily diary that was completed by both participating parents and the child. They did it on the computer at the time. So this was sort of right before smartphones really came of age. They would log in on the website and fill out a number of items asking about parent-child interactions, marital interactions, positive and negative mood, feeling stressed and overwhelmed. And they did this every day for the 8 weeks. Our worst compliance was 21 days. So even the least compliant individuals actually gave us a fair amount of data. And then we had numerous questionnaire measures, and I'll talk about some of those in a little bit. At the end, we brought the children in particular in, but also participating parents. They came in and they did the Trier social stress test, and they also provided a subsample of children and parents provided us with blood samples, basically the ones who were willing to do so could do so. We didn't require them to do it as part of participating in the study. Okay, so one of the, if you want to know what we measured in this study, there's a number of papers that have been published now, but if you want to know like basically every single family environment measure that we looked at, go look at this paper, because we actually used all that data in informing the questions that we had. So this is a paper that was published in Development in Psychopathology looking at gene expression. And so just by way of background, you know, I already showed you this little diagram of gene transcription, particularly pro-inflammatory genes. And the key thing here that I want to highlight is, you know, there are these transcription factors that when they bind to the genome, they promote the transcription of genes. And we can use patterns of gene expression such as the expression of genes that have response elements for NF-kappa B or the glucocorticoid receptor as windows into potential activity of those transcription factors. And what a number of folks have triangulated on, your former colleagues, Greg Miller and Youth Chen, that social adversities like being a caregiver of brain cancer patients, chronic interpersonal stress, low maternal warmth if you've had low childhood SES, that upregulates the expression of genes that have response elements for NF-kappa B and downregulates genes that have response elements for the glucocorticoid receptor. And the idea here is that this could be part of a pro-inflammatory phenotype that might have been something that, you know, we evolved in response to social adversity. And the result might be, you know, promoting more of an inflammatory state when you know, you're facing an infectious illness or stimulus. Now, you know, you can see the social adversities there. What we were interested in was whether we could observe similar associations with mild to moderate levels of conflict in our families, and also with levels of warmth and support in the families. And so we looked at both of those constructs with, again, all the data that we had available. We had this construct assessed through the interviews, through a number of questionnaires, which I'm not going to be able to get into detail, and through the daily diaries. And we used all of that available data to create composite family conflict scores. So for example, for a given set, this could be like a mother and this could be a child, we standardize these scores within mothers and within children. And we took the average of, say, warmth-related measures for interview measures for parents on the diary and on the questionnaires to get a composite for the parent, a composite for the child. Interestingly, these were not that correlated with each other. I mean, they were usually positively correlated, but it wasn't super strong. I mean, I want to say maybe like in the 0.2-ish range at best. But, you know, each person is part of the family. That's unique information that they provide. And so we actually then combine those scores. So even though they're not that highly correlated, they do provide unique information, which we wanted to make sure to capture. So we had a composite for warmth. We also had a composite for all the conflict-related measures. And what we could do is we could then compare gene expression between, let's say, for the children in the sample, the children who were in families with high levels of conflict versus the children in families with low levels of conflict. What were the differential patterns of gene expression across, I guess in this case, 47,000 genes that we looked at? So we could do this by comparing, you know, high and low warmth and looking at gene expression there. This is just a picture of the array, and then high and low conflict. I should mention that warmth and conflict were highly correlated. So the absolute value of the association was about a 0.75. So those were strongly correlated. And we actually, given that, we still did look at them together in some of our analyses. And the pattern becomes kind of interesting to see who ends up winning, the unique contribution of warmth or the unique contribution of conflict. So what we did is we had these 47,000 genes. We looked at which genes had a 1.2-fold difference between, say, high and low conflict families or high and low warmth families. And that gets you about a list of 300-ish genes that were differentially expressed. And then what we could do is sort of take those genes and say, okay, what are the promoter regions on those genes? And of the ones that are there, you know, which of those are sort of NF-kappa B related and which are glucocorticoid receptor related? And so these are the results for warmth. So here we're going to, let me just orient you to the figure. I'm going to show you kind of bar graphs for comparing children who are high versus low on warmth. So these are behaviors that are like spending time together, being involved, satisfaction, and so on. And then this is going to be for the parents. And then I'm going to show you one set of bars that a set of bars for the genes that have response elements to NF-kappa B. So you can think of this as kind of pro-inflammatory gene expression. And then here we're going to have a set of bars for the glucocorticoid receptor. So that's kind of like how much is there a difference in the sort of anti-inflammatory gene expression there. So for the children, what we found is that children who were high in warmth compared to children who were in families that were high in warmth, compared to children in families that were low in warmth, they had lower expression of genes that had response elements to this pro-inflammatory transcription factor. For the parents, you see something similar, but that's not statistically significant. Although when you include conflict in the model, and this is in the paper, this actually, you do see that this goes to statistical significance. But for the children, we see it even without looking at conflict at the same time. So this at least suggests one way that social buffering might operate, at least in children, which is that children in family environments where there is more support and warmth, their immune cells might have just sort of lower expression of these genes that respond to the pro-inflammatory transcription factor. Same thing, but now for conflict. Here are conflicts defined as behaviors that include arguments. These include like self-reports of if the child saw fighting or if the parents also reported that as well. And here what we found is that in children, being in a family that where you see higher reports of family conflict, you see greater expression of genes with response elements to NF-kappa B. And for parents, you see a similar pattern as well as higher conflict associated with lower expression of genes with response elements to the glucocorticoid receptor. So this is actually kind of like what you would sort of expect to see in the sort of pro-inflammatory phenotype, which is, this pattern. I didn't mention covariates, but they're listed here for each of the different populations. Okay. So when it comes to these pro-inflammatory signaling mechanisms, the patterns of gene expression suggest something about the potential for the immune response, what might happen, where high family conflict is related to a pro-inflammatory phenotype, and to a lesser degree, high warmth being related to a less pro-inflammatory phenotype. One of the interesting things is that when you look at the cell types that are involved, and there's lots of immune cells, where we see these patterns of differential expression are primarily, like they're the cells that respond to infection and injury, macrophages, dendritic cells. They're the ones that are like some of the first responders. So what does that mean about potential health implications? Well, recall that we designed this study to look at susceptibility and severity of upper respiratory infections. The reason that the diary was so long, that it was 8 weeks, was we wanted to try to catch kids getting sick naturally. So we only ran the study during kind of the fall and then also during the winter, and then we didn't run anybody past, I want to say, like early April. Like we never ran anybody over the summer. Turned out we weren't as successful, interestingly enough, in picking up upper respiratory infections. This was done about like 2009, I think, is when we first went in the field. So this is in the pre-COVID days. But nonetheless, as you'll see, while we got upper respiratory infections, I was hoping to get more sick kids, I guess. So here's how we did this. We, every day for the 56 days, children reported symptoms of upper respiratory infection on the daily diaries. So did they have a cough that day? Did they have a runny nose? Did they have a stuffy nose, sneezing, etc.? And the important part about all those symptoms is that inflammation underlies all of those symptoms. So think of every single symptom you've ever had if you were sick, in the last couple of months with an infectious, with a cold or the flu or COVID. All of those symptoms are due to the effects of inflammation on the sort of surrounding tissues. So we had algorithms that could kind of alert us whether a child was sick or potentially sick with an upper respiratory infection if they had some combination of, I think, cough, stuffy nose, and I think sneezing for like 2 days in a row, then we would call the family and just sort of check in to see if they think their kid is sick. Parents could also call us as well. And importantly, if they were, we would send a nursing student typically who is trained on doing a physical exam to look in their ears, look up their noses, look down their throats to see whether those symptoms were consistent with an upper respiratory infection like the cold or flu, or like an allergy. In that case, it's not a verified episode. So this is an actual kid, even though they had sort of these elevated symptoms this particular day, for this time, when we sent somebody out, we could verify, yes, indeed, this was an upper respiratory infection. We could rule out other causes. So for each child, we determined the length of the episode. So this might be a 1, 2, 3, 4, 5-day episode, the average number of symptoms during that episode. And then for each kid, we determined the average number of symptoms on non-upper respiratory infection days. So like this and this, the average of these things together, minus, whatever, or not including what's in this shaded bar here. And we did this with all children, regardless of whether they provided us with a blood draw. And here, what I'm showing you is sort of an example of a couple of graphs. So one thing to note is on the x-axis, this is family warmth. So higher numbers mean more warmth in the family. The negative numbers mean sort of lower warmth in the family. This is the average number of upper respiratory symptoms per day. This is all the 59 children in our study, again, the participating child and some siblings. And this is the average on days when they had no upper respiratory infections. And kind of not surprisingly, well, I mean, they're not sick, so there's not a lot of inflammation going on that day. I just put this here. This becomes relevant for the next graph. The circles represent kids who didn't provide us with a blood draw, and the triangles represent kids who did provide us with a blood draw. So the thing that I was sad about with this study is of the 59 children, only 12 of them had verified upper respiratory infection episodes. And if you have kids in this age range, I mean, I guess it could be worse. It's certainly better than what things were like when kids were like 3, 4, 5, or 2. But I was hoping for more. But oh well, this is what we got. Because of this, I like to think of this finding as the sort of, those like results from like really early day fMRI studies where you only had like 5 subjects or 10 subjects. So this is kind of like that. So when you see the effect size, kind of ignore it, focus on the direction. And so what we found is that higher warmth in the family was related to less severe upper respiratory infection symptoms when children were actually sick. We even see this after controlling for negative mood, because negative mood is something that we know is related to the severity of upper respiratory infection symptoms. And we also see the same effect for conflict. So conflict it's just the opposite direction. So higher conflict during periods when kids are actually sick, they have worse symptoms. So this at least provides some suggestion that when for kids in these low warmth, high conflict families, so again, these are the low warmth families, they have higher, more severe symptoms, maybe this is the pro-inflammatory phenotype in action. Okay, so that is sort of our work on pro-inflammatory signaling, which we did in kids and adults. I'm now going to turn to relationships in adults, focusing on the adults in this sample. So here I'm going to talk about gene expression as well. This is work that was done collaboratively with Jude Carroll, who is at the Cousin Center for Psychoneuroimmunology. She's our sort of local UCLA expert at all things related to biological aging. And then this is Kelly Rencher, who is she was a postdoc working with us. She's now on the faculty at the Medical College of Wisconsin. And what we were all interested in was cellular aging and how that's related to stress and social relationships. Now, when I started working with Jude back in 2010, one of the first things she said to me when I started to express an interest in cellular aging is, well, you need to read this paper. And the paper she's talking about is this one. And it's one I highly recommend if you're interested at all in cellular aging. It's called The Hallmarks of Aging. And in that paper, they describe the biological kind of indicators of aging cells. And here's those indicators. Things like genomic instability, things like telomere attrition, epigenetic alterations. I'm going to focus specifically on this and cellular senescence, and here you see a host of them. And, there's a lot of researchers doing research in these areas now, like mitochondrial dysfunction and Martin Picard. So lots of work that's actively happening. Excuse me. So my focus is on these sets of measures because we were able to look at telomere attrition and epigenetic alterations in DNA. This is a blood DNA tube. And then we looked at mRNA expression for this gene called it should say CDKN2A, sorry. It codes for this marker expressed in cells that are old. So if you imagine every single cell in your body, it goes through a number of cell divisions. And after the 50th or 100th division, those cells are much older now because they've gone through a lot of replications. And they start to express more P16, in part because as these cells get older, they don't work as well anymore. And P16 helps stop old cells from continuing to divide. That's why it's viewed as a marker of cellular senescence. Because older cells, they don't work as well. They actually are more pro-inflammatory, for instance. And there's a whole field in biological aging where they are interested in, for instance, using pharmacological agents to get rid of senescent cells in your body and thereby making you younger, so to speak. Levels of this marker are correlated with chronological age in large studies. This plays a role or this is activated when cells are hit by oxidative stress, that is like these free radicals that damage DNA and so on. So these are the markers that we're going to be looking at. Now, one of the big interests, of course, now like 20 years ago from Melissa Eppel's work on stress and telomeres is the experience of stress, are one's appraisals of stress systematically related to biological aging, irrespective of chronological age. We wanted to do that with this marker in particular, because no one had actually done that before. And so in our adults, we had 73 adults who gave us blood from our study. And we had 3 different types of stress measures. So one is the UCLA life stress interview. We had exposure to chronic stressors in a number of domains. And higher scores mean kind of more adversity. This is like the average was about a 2. A 3 would be having significant problems in 1 to 2 areas out of like 1, 2, 3, 4, 5. But we don't have severely stressed folks in the sample, so I just want to point that out. Here we have the PSS score. So these are smooshed histograms, and you'll see why in a second. But I wanted to show you the distribution of each of the measures. This is the perceived stress scale, rating feeling stressed, upset, angry. Our average was about a 12. That's about a third of the standard deviation below national norms and below the original Eppel sample. So again, like these folks are not as stressed as like Alyssa's original caregivers were. And finally, on the daily diaries, the adults reported when they felt like if they felt stressed or they reported if they felt stressed and overwhelmed on a 1 to 4 scale, where 3 and 4 were mostly or completely, I think accurate, I should say, like stressed, mostly accurate for my day, or completely accurate for my day. And if they reported that 3 or 4, then we counted that as a stressful day. And we totaled that and took a percentage. So this is the percentage of diary days out of the total number of days for each participant that they completed, that they reported that like mostly or completely stressed. And it was about 1 in 5 days on average, so 20%. These are all correlated, not surprisingly, about 0.4, 0.5. We saw no associations with telomere length, which was surprising. But given that these are less stressed samples compared to some of the other work, maybe it's not. We looked at epigenetic aging, similar direction, more stress, more aging, but those were also marginal. What we were happy to see, though, was for P16 across both measures. So I showed you the x-axis. On the y-axis is expression of the gene that codes for this cellular senescence marker. And across all of the types of measures of stress, higher self-reported chronic stress exposure, perceived stress, and higher, more days that you report as being stressful were related to higher expression of this marker that indicates cellular senescence. You can see the kind of R squareds there. If you put these all in a model together and pit them against each other, The winner is actually perceived stress, which I think is really interesting, that the appraisals really were the one that sort of take up most of the variance in the association. Yeah, and these two no longer they're no longer significant. So this was really exciting for us because this was the first human study to look at expression of this cellular senescence marker. And so in addition to being excited about that, we also wanted to look at like the social buffering piece. So could social buffering weaken the association between stress and biological aging? So this was actually a core interest of my postdoc and colleague Kelly Rencher. She was at the University of Arizona, strong interests in communal coping and we-ness in couples, kind of defined as a sense of closeness in relationships. One way to operationalize that is by measuring we talk. This is a nice review by a student of Megan Robbins. And this is just an example. This is like the UCLA fight song. You measure the number of first person plural pronouns in a transcript, for example, and see how that might be related to whatever your outcomes are. We looked at word use in the life stress interview transcript, so it's a very different context than, say, talking about a problem in your relationship. And so not surprisingly, we didn't see anything with this particular measure and any of the outcomes that I talked about. But another way to look at this is by kind of asking people to rate kind of how close and how much do they think they overlap with their partner. And so in our sample, we looked at our iOS measure. So many of you might be familiar with this. You have these two circles, and here it sort of represents greater closeness or not very much closeness at all. So what we did with this sample is this is the same plot of P16 and perceived stress scale score. And now I'm going to sort of split it off into three just for display purposes. So in the middle here are folks who reported a 3 on the iOS. So this is like how much overlap they had in their circles. They looked, and so again, higher PSS, higher P16 expression. For 0 to 2, kind of the same thing, higher PSS, higher P16 expression. And then interestingly, here for the folks who reported more closeness, there wasn't an association between the PSS and P16. So suggesting that might be consistent with this social buffering idea. If you want to learn more, and there's some really interesting gene transcription work and as well, this is the paper published in Aging. So we're really excited about this work. Like it allows us to add to the list. So up until recently, it was really just telomeres. Now more so epigenetic alterations, but now looking at cellular senescence is I think we can add that to the list of things that would be of real interest if you're interested in the intersection between psychosocial factors and biological aging. Okay, so I want to end by turning to implications of this idea that social buffering might have these effects on inflammation, for example, for other types of exposures. And so back to our cells, the immune cell responds to things like bacteria. Another thing it can respond to are the stuff you breathe or consume, so pollutants. And this is highly relevant in the context of the air that I walk around in every day. So this is a picture of Los Angeles from Griffith Park. This is you can see it's a little hazy, right? This is smog. And the stuff that makes up smog and pollution, number of things, nitrogen oxides from fossil fuel combustion, the small amount of traffic that we have here, for example, but also energy production, industrial processes. When those nitrogen oxides combine with other compounds in the presence of sunlight, that forms ozone, also not great. Particulate matter as well, so from combustion and other sources, is another thing that I get to breathe in every day. And UCLA ranks #1 in various things here in the US, but we're not terribly proud of having the Los Angeles region ranking #1 in ozone pollution and #5 in particle pollution, because that's really bad and it's harmful for people with respiratory illness. And I'm going to focus specifically on asthma. I got interested in this about 10 years ago when I was having a conversation with an epidemiologist at USC who studies this work, and I'll sort of highlight that. So these gases and particles, they make our way to our lungs. They're then encountered by cells like macrophages and dendritic cells. Those are the same cells that show the differential gene expression that's associated with the family environment. So for instance, like particulate matter can bind to receptors on macrophages. So normally, these cells actually play a role in controlling the allergic inflammation that is part of the underlying pathophysiology of asthma. But when they're activated, that actually stimulates inflammation. It stimulates, or allergic inflammation in particular. It stimulates airway obstruction and hyperresponsiveness that you see in asthma. And so I was really interested, given that these are the cells that were sort of responsive to the family environment, could we look at how the family environment might be related to, like symptoms of asthma in the context of both, in the context of pollution? And there was some suggestion that that might be an interesting question. So these are studies that were done in the LA region by folks over at USC. This is the USC Children's Health Study. They had over 1,000 children. And in this first study published in PNAS, what they found is in families who reported high perceived stress, greater exposure to traffic-related air pollution was related to higher likelihood of new onset asthma, and then worse, in another study, this one right here, worse lung function, objectively assessed lung function. Another research group in epidemiology here, they looked at in homes where there was no father present, what they found was greater exposure to air pollution was related to worse lung function. They also looked at fighting in the home. So they had this one item, people in my family fight a lot, true versus sometimes true or not true. The effect was in the same direction, not statistically significant. These are not like the greatest measures of the social environment at home, but that at least provided an initial signal that there might be something interesting there. So the question we asked was, could family support at home buffer against the harmful effects of pollution exposure? So this is work that was done collaboratively with Sunny Bay, who is actually, she was a postdoc at the time. She was also a grad student working with me in RENA. She is now an assistant professor at the New Balmer Institute in the University of Oregon, a really cool institute looking at children's health, children's mental health. And also Yingying Meng, who is a senior research scientist in public health. In California, there is a biennial California health interview study. It's really designed kind of more for health services reasons primarily, but it's been used in a lot of other ways as well. And we were able to access data from the 2003 survey, and we looked at adolescents between the ages of 12 and 17. And I'll tell you why 2003 is important in a second, but based on that previous data and some of the other literature that I looked at, what we predicted was that having low support at home, so like being over here on, say, this part of the perceived support from an adult at home, in the context of high air pollution exposure, should be related to worse asthma severity, if this whole pro-inflammatory phenotype being responsive to the family environment idea is true. It's really not clear what to expect for low pollution exposure. In general, you don't see a lot of main effects for pollution exposure, for support or stress on asthma severity by itself. The effect of pollution exposure on asthma severity is also kind of small. So I didn't really have any strong hypotheses about what was going to happen with low pollution exposure. Okay, so how did we measure air pollution? So Yingying Meng, she was able to harmonize children's health interview study data with data from the California Air Resources Board from their monitoring stations. So they have monitoring stations throughout the state, and they're good for some things, not for others. For gases like nitrogen oxides and particulate matter, it's actually not great for that. You really want much more precise estimates, and the data that we had for that doesn't really enable that. But for ozone, which has lower spatial variability, it's reasonable. And so this is LA County, and here's where all the monitors were. And so what we did is we tried to kind of look at adolescents in the sample who had lived within 5 miles of one of these monitoring stations. Now, this is all over the state, but I'm just highlighting LA County here. So here's the demographics. So, we have about 30% Latina, 35% white, and you can see the other race ethnicity groups here. half female, 14. About 1 in 5 were at 0 to 99% of poverty level. Okay, so why 2003? So the California Health Interview Study it was annual and then it became biannual. It happens that in this 1 year where Dr. Meng had harmonized air monitoring results with the California Health Interview Survey, they happened to ask these questions in this particular year. This is the only year that these questions have ever been asked in the entire history of the California Health Interview Survey. And so it was really kind of serendipitous, at least, that we could kind of take advantage of that. As I learned later, these are actually administered fairly frequently in, actually, I fill out this California parents of kids in public schools end up filling out these particular questions like all the time. But they happen to only include it in this particular year. So that was interesting. If you look at the items, at least to me as maybe someone looking at it through a social support lens, these look like perceived support items to me. So that's kind of how I interpreted the construct. And then on the y-axis of the graph that I'm going to show you in a second, we had asthma severity measures, or a single item measure of asthma severity. During the past 12 months, how often have you had asthma symptoms such as coughing, wheezing, etc.? And again, these are all kids who do have, at least they report that they have a diagnosis of asthma. So this is symptom frequency, not at all, once a month, every month, and so on. So what we were really interested in was basically a pollutant by support interaction, because that's kind of the key test of this buffering hypothesis. And we found like a little bit of signal. I mean, it's intriguing, but like the overall ozone by support interaction, it was just like right at 0.049. It does go away when you adjust for covariates, but I just want to show you what it looks like. So for folks with sort of average or below average ozone exposure in the blue, these are the associations between perceived support from an adult at home and asthma symptom frequency. For the folks exposed to higher ozone levels, you see that lower support is related to higher frequency, higher support related to lower frequency. So, there's some evidence here for this kind of buffering idea. There's a lot of limitations to this work. Part of it is the measurement of pollution and the measures of family environment, at least in other studies. But at least it suggests a direction of interest, which is that perhaps in the future, studies that measure the family environment, look at personal pollution exposures, and look at clinical endpoints, maybe there is something there for social buffering. Okay, so I just want to end where I began, at least this part of the talk, which is, there have been systematic scientific approaches to looking at psychosocial factors and how they influence health and illness. In terms of the ideas about social support buffering those effects, you can see those here. And since that time, we've made a lot of strides in understanding social environments and how those environments are processed by the brain and transduced into the cell. And what some of this work suggests is that, pathways like pro-inflammatory signaling and immune cell aging might be ways in which that social buffering occurs. And then most importantly, we think of the stressor as the thing we're buffering against. And what I want to impress upon you is that things like infections, things like pollution might be another thing that the social environment helps kind of protect us from in some way. And so just another reason why cultivating healthy family and intimate relationships might be important for our health. All right, so I'm going to take a breath there for a second, let you think. If you have questions, maybe what you might want to do is just sort of write them down somewhere. And then is it okay if I just sort of do the second part? It's pretty quick. Okay, great. Okay, so part 2 is much shorter, but I did want to share kind of things that we're doing on campus, which I think might be of interest to some of you. Let me go here. Okay. All right, campuses to communities. So as Francis talked about, we've had this review article in American Psychologist. Julianne Holt-Lundstad was the first author, and then I worked on it with her and David Sabara. We were kind of like put together. Well, we all knew each other and were friends, but we were put together in part because we each had done different meta-analyses looking at some domain of social relationships and how that's related to health. And so they asked us to put our heads together and just write a review. And so in that process, we were like, you know what? like there's so much evidence here. You know, if you think about, for those of you who are kind of not as, those of you who are not young like myself, you'll remember like House, Landis, and Umberson in Science in 1988. And I thought that that would lead to the kind of things that we're seeing now, but it didn't. And so we felt it was important to send the message that advancing social connection as a public health priority is an important thing. The person who has really taken all of this work up, I should say, is Julianne. This is a picture of her actually testifying in 2017 at a special Senate Committee on Aging. And of course, I think all of you now know, this is just some of the conclusions were like, make it a public health priority. But most of you know from last year, you know, the culmination of this work and really Julianne's, because she actually helped write this report, is the US Surgeon General's advisory on social isolation, social connection, and loneliness. And if you haven't read it, I highly recommend it. I know some of you have. So around the same time, in spring 2017, my colleague, Chris Dunkelstetter, you probably know her, she's a distinguished health psychologist. She mentioned to me that she was working with a group called, at the time, the UCLA Healthy Campus Initiative. This was started in 2013. It was envisioned and philanthropically supported by Jane and Terry Semel. Terry Semel was an executive at Warner Brothers and CEO of Yahoo in the early 2000s. I think, unfortunately, now has dementia. The family they were major supporters of mental health research and clinical care. And in 2018, they actually endowed it. And so that's why we call it the Semel Healthy Campus Initiative Center. I'm going to say Healthy Campus for short. The very bold vision is to make UCLA the healthiest community to work, learn, and thrive, and to inspire others both locally and beyond. I was trying to think of a way to describe this to you, and I think the best way to describe like what this is to describe the very first meeting of the steering committee for this group that I attended. So here's who was at that meeting. The person in charge of it, she's an associate vice chancellor who's a pediatrician by training. Our provost, our vice chancellor for student affairs, deans from multiple colleges, undergraduates involved in health-related service and leadership positions, our campus sustainability coordinator, our executive director for events and transportation, our director of recreation, our director of our student health center, our director of our student counseling center, the person in charge of housing and hospitality services, so the dorms and dining, faculty in public health, grad students in public health, faculty in psychology and psychiatry. And what's really funny is like we had a meeting yesterday, and I think almost all of those same titles were basically there, which is kind of neat. So basically faculty, administrators, staff, students who all have some stakes in making sure our campus is as healthy as it can be. So the way this works is it's organized into 6 pods, and there's leadership from a faculty member and a staff person. If you want to learn more, because I'm not going to give you like a whole overview, you can see the website there. But I did want to talk about how it's an example for being a spark plug for improving health on campus. And the best example is the Eat Well pod. I'm super jealous of this pod. I wish we could do a fraction of what they've been able to do in the last 10 years. It's co-led by a professor in integrative biology and physiology, and the person in charge of operating the dorms and dining facilities and conference and hotel facilities. A number of things that they've done. They were able to change the placement of healthier options so that they're eye level in vending machines. We didn't exactly phase out sugar sweetened beverages yet, but they're like at the very bottom of the soda machines. It's like all water right here. We learned through their work that food insecurity is a problem on our campus. So in 2016, university surveys found that 37% of undergraduate students were food insecure. And they helped support and promote a number of programs across campus to help meet basic needs. That number is now 27%, and there's still a lot of inequities, as you can kind of see here. So we still have a long way to go, but they've made progress. They created a food studies minor. And then just before the pandemic started, our recreation department opened up a teaching kitchen where to help improve food literacy and build community on campus. And actually we have 2 teaching kitchens now. This is our outgoing chancellor. So that's just a sample of what they've been able to do over the last 8 years related to food. So in 2017, my colleague Chris said, maybe we should, she talked to their leadership because she was participating in the Mind Well pod at the time. She said, you know what? there's all this data on social connections and health. Maybe we should start one up, a pod related to that. So that led to the creation of the Engage Well pod. We've been doing that now since 2017. My colleague Chris is now retired, so it's actually me and my colleague Aaron McMahon who runs our recreation department. These are our goals, what we're hoping to do. We meet monthly, well, actually now we meet quarterly, staff, faculty from all over campus and so on. Just to give you a few examples of what we've been able to do in the brief time, and again, it like pales in comparison to the Eat Well pod. Here are some of the accomplishments we did before COVID. One of the things we did was we wanted to promote the importance of social relationships for health and just kind of communicating with others in general, as well as social belonging. Some good examples of that are, if you're familiar with Art Aaron's Fast Friends paradigm, the 36 questions to love, if you look it up in the New York Times, we created cards that we would hand out at some of our programming and other people's programming to encourage those kinds of questions. Chris kind of single-handedly helped kind of start this kind of messaging on our campus around being welcoming to all sorts of identities. She also she had some initiatives and actually continues some initiatives to improve mentoring on campus between faculty and students and faculty and faculty. One thing we can't take credit for is that, but the HCI can, is they brought the diabetes prevention program which is this curriculum that has been effective in reducing type 2 diabetes risk in pre-diabetic persons. They brought that to UCLA and piloted it here. And then at a meeting, the director of the dorms and dining said, wow, like you can reduce like type 2 diabetes by this much, or that can. I would like to provide that for my employees, many of whom are service workers, many of whom, low income, so you would be worried about type 2 diabetes risk in those folks. And he provided it to them on the clock and is still doing that. And so one of the things that we did, one of the very first things we did was we were really interested, for instance, in like, what's the social consequences or correlates of participating in a lifestyle management program with your fellow employees? And do changes in your social relationships, could they be related to outcomes in that program? Surprisingly, nobody has tested anything social in these diabetes prevention-like programs. And so this is actually a paper that we have under review. I'm just going to briefly describe the design. So these were 79 staff who participated in this unit-sponsored program. This was at the time in person. They were followed over 5 waves, 1, 3, 5.5, 8, and 11 months later. This is in bold to remind me that this was when the active 15-week curriculum was going on. They're doing classes learning about exercise and diet. And so we had sort of, this is just for display purposes, but just to show you, we measured the ISL, the interpersonal support evaluation list, a measure of perceived support. In some folks, their perceived support went down during the time that they were in the study participating. In some folks, it stayed kind of, it didn't really move that much. And in some folks, it went up. Now, this is a single trial design. This is actually like in the real world. We don't have a control group or anything like that. So just kind of keep that in mind. But what was interesting about this is we could look at the association between changes in social support while participating in this program and outcomes like weight and physical activity. And the main thing, it's really hard to see, but basically one of the primary findings is that individuals who showed an increase in perceived support during the time that they were in the intervention, they showed the largest weight loss. And it was the reverse for the folks who showed a decrease in perceived support. Now, this is all correlational, single arm, but first time anybody really looked at this looked at the association between outcomes of the diabetes prevention program and changes in someone's social network. And interestingly, this was mediated by self-esteem, by self-efficacy. I'm sorry. So the folks, one of the explanatory factors was an increase in self-efficacy. I'll close by just sort of 2 things. So then COVID happened. We had all these ideas, like one had to do with like getting strangers in the dorm dining halls to kind of like meet and talk together. But then COVID happened. So some of the stuff we did during the time were I'm sure your campus had lots of messages going out over like how to cope during this time. And we made it a point to include in that messaging things like social distancing does not mean social isolation. Reach out to other people. Do things that give you a sense of purpose and meaning. Those were all things that we sort of intentionally had inserted into communications about health and well-being. My colleague Chris noted that one of her grad students was really isolated, and she lived in campus housing. And so she got leadership in our graduate school to get together with the housing folks, and they started to meet monthly to discuss outreach to grad students during 2020. One of my favorite examples is that because of that, they created a farmer's market distributing food and baked goods from our campus commissary directly to graduate students. And for a long time during the pandemic, they met bi-weekly. We did some podcasts, you can listen to them. Last thing I'll mention is that, of course, during 2020, we had all the concerns renewed about racial justice. And so one of the things we did was we surveyed our staff and faculty about the prevalence of experiences of microaggressions. And so this is actually this is data from that. You can see, for instance, that women reported higher, more experience with these are female staff reporting higher experiences with microaggressions compared to men, African Americans more than whites, and so on and so forth. Multiple disabilities reporting more microaggressions compared to folks with no disabilities. So I'm going to end by just leaving you with where we're at and what's on my mind around all of this. I'm going to give you sort of a physical metaphor. So here's where we're at. This is a major intersection on our campus. It's Westwood and Strathmore. It's right in the heart of campus. This was taken when I started at UCLA in 2007. There's a pod that focuses on the built environment. And they shaped this intersection to look like this. It's much more pedestrian and bike friendly. It's got those diagonal crosses where everybody crosses at once, for example. They made physical changes to the environment. You can see the green bike lanes. Structural changes. They reduced our campus speed limit to 20 miles per hour. It's made our campus safer, made it much more physically active. One of the big questions we're trying to grapple with is what is the equivalent of this for promoting social connections and healthy relationships at an institution? It's been a challenge. I'll mention one sort of reason why it's been a challenge. But one of the other things I'll mention is here's our sort of priorities right now. We're figuring out how can we increase feelings of belongingness. I want to assess what belongingness looks like, and for the folks who feel like they don't belong, what are they doing? And then how can we cultivate social connections sort of through pro-social behavior? Steve Cole's working with us on this, and it's a big interest of his. All right, last thing, and then I'm done, sorry. Health and well-being are not just the territory of student health centers or staff and faculty wellness programs, and that's why a lot of this got created. But one of the big challenges we face is if you look at any of these domains, the built environment, eating, moving, mental health, and you think about who on your campus is responsible for those things, you can probably think of who those people are, right? Student mental health center director, the person in charge of dining. But for improving social relationships on campus, we don't exactly have a point person or administrator for that. And that's been one of the main challenges that we've been facing. But it's sort of a fun challenge to tackle, and it's led to all sorts of ways to kind of interface with the campus in ways that I had never been able to do so before. So it's been incredibly fulfilling, but also super challenging. So I will stop there. I know I've been talking for a bit, so but thank you for your attention. Welcome to take any questions around sort of part one or part two. And again, thanks for being here today. Take this off. Share. Okay, great. All right, happy to take questions. Yeah, Anita.
Great talk, Ted. Thanks so much. It's really great to hear an update on what you're up to. We know social relationships are important, obviously, for so long now. And as you said, the Umberson paper came out and Jim House so long ago. It seems like we've got, looking at your work and others' work, the biological pathway and the health behavior pathway. And like just thinking about the asthma work you were talking about, what do you think is the core component here? Is it that these warm, supportive families are better at engaging in appropriate health behaviors, or that there are under the skin type of changes biologically? And can you just speak to
Yeah,
those, how policymakers, I mean, you're at that point where it's like
right.
about, what do we do now? Where do we put our efforts?
Yeah. I think I did this review recently on social relationships and the immune system in children, and that was a really fun one to do because I got to kind of grapple with these kinds of questions. And one of the things I came away with is I kind of think it's some combination of what can promote a sense of safety. And this is not my idea; it's not new. I mean, there's a number of folks who have had this idea. Like it's like safety and certainty, and then making sure everything else around you is like not harmful. But if you don't feel safe and you don't feel, if there's a lot of unpredictability because of your social environment and stuff, and you're surrounded by, you name it in the biological threat category, whether that's pollution or bacteria or whatever, like that is not a good combination. We don't know to what degree feeling safety and like predictability is going to protect you from like the really bad things that are out there in terms of these biological exposures. So it's sort of like whatever policies and changes need to happen for kids, for adults, to promote, and obviously the social piece is a huge part of that in terms of what are our cues for a predictable environment. It's like whatever can do that might be the kind of the route that we take. The problem is that that's a really broad set of things promoting, how do you make a child feel safe in this world? It requires a whole lot of things, right? How do you make an adult feel safe at work, in their relationships? There's so many touch points for that. That's sort of what I've settled on in some ways.
That's key. I've got a couple of questions, but I'm going to let other people do theirs, and then I'll come back to you if there's time.
Sure. Yeah.
Maybe I should.
I'll ask. I have a few too, so I'll go with it. Yeah, Ted, so inspirational. I'm just really excited about all the work you're doing, both part one and part two.
Yeah.
I have, let's see, a question about like part two. Well, I have one more specific question I'll start with, and then a broader question. More specific question, you mentioned something intriguing. You said in the diabetes prevention program that the role of social support is like actually mediated by
self-efficacy.
Self-efficacy, right? Can you break that down? Like that was intriguing to me because I would have I don't think I would have necessarily predicted that, right? Like the
Right.
ISCL is asking about like how much do you have in your environment? Like, do you have someone who can help you if you're in trouble and things like that, right? That's what I have in my mind. So how would having higher levels of that, like, then sort of, would it then, like, I'm just really curious, like how that then translates into self-efficacy.
And the other interesting thing about it is that so one of the things we've gotten critiques on for this work, which I think is fair, is, if you study, like for the folks who study lifestyle interventions, like diet and exercise interventions, one of the things that is important is like your self-efficacy around the thing you're trying to change. So self-efficacy for exercise, self-efficacy for making diet and nutritional changes. And we did not actually ask that. And so this was actually sort of in general. And, it's hard to know, again, because of the design, right? Because, if you keep in mind that like for the first two waves, these folks are in like weekly classes where they are like their progress is being monitored in terms of exercise, where their weight's being monitored. They are learning new things. Among those things is learning social support and how it's important, and also learning things that are probably that might actually increase self-efficacy in some way, whether that's like, in week 3, I talk about how I'm having a hard time with this. People give me suggestions. And then in week 4 or 5, I show improvements, like my exercise numbers are much higher up. So I will say it is harder, like the design doesn't really help us with that, except to say that, believing that there might be these sort of like, spiraling effects, I might say, from like, kind of increasingly believing that there are people there who can help you. Like they may have made use of those people. And if they have done that, perhaps then they are doing the things they need to do. And then perhaps in doing so, they are starting to feel like they can do them. But yeah, the design in some ways is too coarse to be able to answer that question. But I think that's where like, I think of like intensive repeated measures, which I know many of you are, do. Like I think of that as if we did that, on a, you can imagine burst designs where you do that for a week and then you do it for another week. That might be able to disentangle that in some way.
Yeah, that makes a lot of sense. Yeah, thanks. My broader question was just I started to get curious when you were talking about the Healthy Campus Initiative. What other, like, okay, I guess a general question is how much is the focus kind of on implementing strategies that are kind of already well established, right? In whatever domain you're trying to influence, eating versus social connection, versus like use this as an opportunity to like, basically how are you collecting metrics on sort of outcomes and like effects of initiatives?
Yeah. So it like sort of broadly speaking, yes. So for instance, there's a research well pod. That's kind of one of the things they do. One of the things that and then there's also, I mean, you probably have an institutional research office at your university. There's all sorts of data that gets collected through that. And so that's a way to kind of look at changes. So for instance, right in the middle of the pandemic, or actually right before the pandemic, we were able to get a few questions that on like perceived social support and loneliness into our sort of undergraduate kind of representative sample student survey. We need to get them back in to see how things have changed. But that at least there's ways that we can look at things at the campus level. And then the other thing I'll say is that there is an interest now in developing like some kind of index for large educational institutions and how healthy they are. And so actually one of the things that this group is working on is trying to develop something that could be sort of exported to other campuses so that they can look at their health as well, whether it's from the policy to the program level, et cetera, et cetera. And then lastly, I think in this sort of social space, it's kind of a challenge because, if you think about like the interventions that we know about or data that we have, relatively small scale, experiments, randomized clinical trials, it's not like you have these sort of regular large social interventions that are then, the folks in public health or other fields are evaluating. So that is a struggle that we've been facing. Like in this sort of pro-social behaviors domain, we've got folks here or like in California who've done like random acts of kindness interventions and things like that. It's one thing to do that. It's another thing to try to scale that a little bit bigger and then measure what's happening. And that's where that's kind of like the space that we're all sort of very new to and not sure how to navigate, but we're trying.
Thanks.
Yeah. Oh, sorry. Jin, go ahead.
Cool. Yeah, thank you so much for the really insightful talk. So just to kind of like piggyback off of Francis's question, it's like really cool to see how many of these like pods UCLA has employed over, the last few years. I wonder though how with like, the initiation of new initiatives or new like, directions or interventions, like how does this translate to like increasing costs for the student for just the university, and how this actually kind of may have like a negative feedback into kind of promoting some of these issues that you're trying to tackle?
Right. Well, so are you thinking fiscal costs or other costs?
I was thinking more so fiscal costs, but I think other costs probably also like play into it.
Yeah, absolutely. Yeah, just want to make sure. So part of what they've tried to do, I'm trying to think of like initiatives. A lot of like support from philanthropy helps because then, it's not something that is coming directly out of like student fee dollars, for instance. And then the involvement of existing like I'll use the word operators. So like dorms and dining or like vending services. So if you think about something like putting the water up higher versus lower, like that is not something that operationally over time is going to end up coming, like they're not going to raise the price of, soda in order to do that. Although, I mean, I guess in the context of sugar sweetened beverages, short of like we haven't gone that route now that I think about it. We haven't gone to like penny per gram of sugar or something like that, because then that could affect students in a negative way. So I threw out a lot of the things, and even like we're super proud of our dining services. We have like, oh, and I think we just have one cookbook with like this crazy healthy food that like it's even intimidating for me to like try to cook or put together. But they've tried to do those kinds of things, healthy choices for students in a way, like they are very conscious of like what the bottom line is like for students. Trying to think of domains where like there could be, I mean, one area is sort of, like in the area of mental health, it's a big challenge. I don't know what things are like at your institution, but we probably have, like, in order to meet the mental health need, we need about double the staff that we actually have. My wife actually works at the counseling center there. And so like, I hear about this. That would be something, for instance, that would be sort of costly. But one of the things that the state of California did recently was they created a pocket of mental health funds, actually, that then they distributed to the campus. And the campus could do what they wanted with them to sort of like figure out ways to improve mental health, and obviously like tertiary prevention, like more people providing therapy is more expensive than other things. So as an example, and I'm not going to use UCLA as an example, but it's one that we want to think about here, is at UC Irvine, one of the things they decided to do with those funds is to hire someone who can help train faculty on ways that they can incorporate well-being practices into their pedagogy, both for themselves and for their students. So for themselves might be like looking at your syllabus, asking you about like what the arc of your class is like, and seeing like when are things most stressful. Are there pedagogies approaches that you can adopt that are both good for learning and also good for you, and then also good for your students? So that's one way they've used the mental health funds. I think the idea might be if you can just sort of, dial down some of the stress anxiety across many students, that might save you some through the side. But those are the kinds of examples that come to mind.
Okay. Oh yeah, no, it's really interesting. Thank you.
Yeah. Yeah. I know there was another question. Yes.
Oh yeah, thank you, Dr. Robles, for the wonderful talk. I had a question about from the first study, like family relationships. I just had a question about the maybe like potential relationship between some kind of like socioeconomic factors and like the relationship style or like parental behavior, because I think sometimes like I want to be like kind and friendly to everyone that I connect with, but just like if you have too many obligations,
Yes.
or like the autonomy you have over your time,
Right.
or like the employment status, like if you work too much, then maybe you have less time. So I was wondering like how much is the effect you're looking at is coming from like social relationship per se, or whether it's coming from the other factors, like other like systemic factors as well.
Right. Yeah, and in that first study, although in the first, well, yeah, in the first study, some of it we can try to get out with covariates, right? But then the other piece of that is like, there's still probably some association between, let's say like work stressors spilling over, for instance, into your personal life. And actually we've actually had some papers that have looked at that question about like how much is there spillover, for instance, between like if you were on days when you report high levels of family stress, what does your on days in which you report a lot of work stress, for example, what are your social relationships with your family look like? So that first study, again, short of like the covariates that we would have used, I'm trying to remember now if like SES was one of them. that might help us a little bit. I do think it is important to point out that like that study also, the range of SES, while it's not on the super high end, it's also not on the super low end either. And so getting a distribution of that is, you know, kind of a perpetual challenge, you know, in this kind of research, especially the sort of biobehavioral stuff, unless you are really, we tried to go to great lengths to try to recruit, you know, sociodemographically and economically diverse sample, but we still face challenges. The folks who are lower income in LA, it's a big pain to get here, for example, so they tend to be underrepresented. I think post-pandemic, there are ways now to make that a little easier, but certainly always going to be something to kind of think about. The sort of role of, you know, economic stressors, right?
Yeah, thank you.
Question. Yeah, Nancy.
Hi, this was an amazing talk. So ever since I read your paper about restorative processes, which I know is
Yeah.
it's been a long time ago now, I've been really curious about your thinking on that and your work on that,
Mhm.
and whether there are any new developments or just like
Yeah.
areas in which you have suggestions for people who are
Right.
really in
Yeah.
restorative biological processes.
Yeah, you know, it's funny because I, gosh, it was, I'm trying to remember when this was. It was like a year or so ago where there was a, I think it was a workshop that was where they wanted to like, they were interested in this question, like basically exactly what your question was. Like, where are things at with restorative processes? Is it still useful to sort of think about? And so it was kind of fun. I mean, I'm sure those of us, again, who are not young, you know, every once in a while you revisit an old paper to see like, you know, is anything that you said kind of potentially relevant or interesting still? So the things that I think I think the things that have ended up still holding true, like the conceptual stuff, all I think, like it all kind of still holds true in a couple of ways. One is like one way that the psychosocial factors could mess with these restorative processes is when you don't have the time or space to actually engage in that. A really good example is like sleep disruption. So we know that sleep has these restorative effects in a variety of ways. And by simply disrupting sleep, and there's way more data that came out on that in the last 13 years or so, we know that sleep disruption, you know, it's not good, and it does have these detrimental effects. So that sort of still holds true. I think one thing that is sort of in terms of the future, there's been less of a historical emphasis on like metabolic and growth pathways. So things like yeah, I mean, I guess like how in the developing child, how these stressors might influence like growth hormone production or other like IGF related pathways. And then the metabolic pathways, I think we're starting to see much more interest in that. And the idea that I think I had at the time was like, you know, stress and other like adversities could disrupt those metabolic pathways. And there's much more work on that now than there was at the time. So my kind of takeaway from revisiting that paper like a year or so ago was I was actually like, oh, okay, like a lot of that is still kind of reasonable. And then I think the only other thing I would mention is like there's been more work on parasympathetic nervous system. This is sort of related to metabolism and growth. There is more work on that than there was, but there could still be more. So I think that's the other area as well, like parasympathetic pathways that could use more empirical attention. Hopefully that kind of answers your question. Yeah.
Oh, yes, thank you.
Yeah, Christiane.
I had a question again on the other end of the spectrum on the kind of broad scale intervention and fostering pro-sociality and social connection. And you brought up this intriguing example of, you know, going through your syllabus with an eye on cutting stress.
Mm-hmm.
You know, reducing stress for learning, reducing stress for everybody who's involved in the enterprise, right? Have you or has someone else thought about using that same approach for fostering social connection in terms of, you know, can we systematically think of ways in which to, I mean, and we know it's good for learning, right? So
Yeah.
in terms of systematically planting
Yeah.
social engagement.
Yeah, that's a great, I mean, that's a really good point. And certainly like, there's definitely far more literature on like cooperative learning, right? And what that does for learning. What I actually don't know is like in that literature, whether there's work on cooperative learning and say like, does it increase feelings of belongingness? Does it reduce, you know, does it increase perceived support? Does it reduce loneliness? So that I actually don't know. And it would be interesting to sort of think about because a lot of that is in the education world and they're not that far away, but they're also not that close either. But that's a really good point. And actually, what you brought up too got me thinking about another thing, which is for us, there's been this tension between like hitting people over the head and saying like, you should go talk to people, right? Like it's good, like, you know, connect with others, get more friends versus trying to get people to do something that creates that anyway without having to tell them that more friends is good. So this is where a lot of the stuff that Steve's been interested in and overlaps certainly with the kind of work on sense of purpose and pro-social behavior is one of the things we're going to try to do is try to get people to sort of home grow program or just things that they do on campus that like have like fulfill a sense of purpose, you know, or pro-social, with the idea that if they do that, and if we just sort of provide a few guide rails on just sort of like make sure to involve other people, et cetera, that you'll get social connection as a result. And that we wouldn't necessarily have to tell people, all right, you guys should do something good for others and make sure that you know, provide emotional support to each other too. Like it'll just sort of happen. But your example is a really good one I hadn't thought of, which I'm now going to perseverate on, which is, could we look at social outcomes of things like cooperative learning, design, that kind of thing?
Yeah, thank you. I mean, at UBC, a lot is happening in that space for physical activity, right? So, you know, interrupting sedentary behavior, and
right in the
you
classroom.
know, you can bring
Yeah.
in a movement coach into a large
Yeah.
lecture class, and you know,
You make it social, right? Yeah.
yeah. And so it depends on the lens you're taking,
Mhm.
and it may be just a small thing that you change, but it'd be a matter of tracking too.
Yeah, right. Yeah. Importantly, kind of back to another question, you know, what are the outcomes of that? That's really important. Yeah. Thanks for the question.
Yeah, thanks for this great presentation. I want to just ask, I mean, I've been working the past couple of years to kind of increase this sociability to think about how public health interventions can kind of work in this space. And we do get a lot of pushback on this idea of increasing sociability, and particularly people thinking about the individual experiences of, you know, those who are highly introverted, socially anxious, or you know, maybe even attachment style, those who are avoidantly or anxiously attached. So regardless of which framework you kind of approach it from, there's this challenge of that some people seem to respond differently to these messages. And I think, you know, I was running some analyses, and you mentioned kind of workplace spillover and that sort of thing. And I was looking at these relationships between work and, you know, and stress, and people who are stressed definitely just spend less time socializing. And I think that's in some ways kind of a defense mechanism. And I think lots of people who are avoidantly attached, and a lot of this is just like personality defense mechanisms. And so I'm curious if there's any evidence that you're aware of that kind of talks about that as a potential moderator. Like, could we do harm in some ways? You know, if you're like really lonely and you've got bad social skills and you put yourself out there and you have a bad social experience, does that just reinforce your sense of inadequacy as a socialite, right? So I don't know, any reflections on that?
Although what that makes me think of is like the Nick Epley work on how we tend to like underrate how helpful people can be to us, right? And that is in and of itself a barrier to doing to doing that. And then I mean it sort of fits in with the like I think of the work example where, you know, you've got for whatever reason you have stress at work. It makes you, I mean, part of what burnout is, you know, feeling detachment from other people at work. And so what the question becomes is like, can there be a way to kind of not have the detachment from others, but also not contribute to, you know, making this much more of a stressful work environment. So, I mean, like what it comes down to in some ways is like, yeah, can you encourage folks to connect with other people, like let's say at the workplace, in a way that doesn't have a side effect of increasing the amount of sort of overwhelming feelings and stress and that kind of thing. You know, are there ways to do that? And that, again, we don't, I mean, there are workplace, there's a workplace intervention that I'm particularly fixated on because it's got like, because it actually, it's essentially an intervention on how to provide social support. And like what the point isn't to like pull somebody aside and then just like start supporting them. It's more like give the supervisor a menu of things that they can do if something comes up. So that way, if someone chooses to say, you know, I'm having a tough day, you know how to respond versus being clueless, you know, that kind of thing. So yeah, I mean, those are my sort of thoughts. It's like, are there ways that you can make the social interaction less stressful while at the and not overwhelming for work, while also giving people the skills they need to be able to be helpful when needed?
Yeah, thanks for that. I as you spoke, it also kind of recalls back to your previous answer about safety, right? Of you know, I kind of think of psychological safety, and
yeah,
I think in the workplace intervention, Terry Beer, you know, at Michigan, has this paper that I thought was really interesting, where they found actually social support in the workplace can actually be harmful from coworkers or supervisor because you feel demeaned or you feel incompetent, you know, in receiving that social support.
Yeah, like in all the ways that social support can be non-helpful. Exactly. Right. Yeah.
Okay. Thank you.
Yep.
Thanks so much, Ted, for the talk and the discussion. I think we're out of time.
Okay.
So I'll stop the recording.
Okay.